Hello, my name is Cole, and today we will talk about metabolomics of giant panda bamboo diet. So I went over the paper, I read the paper, A Diet Diverse in Bamboo Parts is Important for Giant Panda Metabolism and Health, done by these guys. I will not attempt to say their names because I don't want to butcher it. But their study basically focused on this question about how do these pandas get the necessary nutrients they need when all they eat is pretty much bamboo, which is, you look at their name, the giant panda. How do these pandas get so giant in the first place when all they eat is this hard, grassy plant? So, what they believe is that the different parts of the plant are actually key to how they get different nutrients. Each part of the plant may actually give a different amount of nutrients or metabolites that they need to grow and survive. So, I just want to go ahead and show kind of the parts of bamboo in case you didn't know. So this main part that you see, the big column that sends out of the ground, this is the culm. Then they have this sort of seed-like thing that they grow from. This is what's known as a shoot. And then there is a third part that the scientists focus on, which is their leaves that will sprout from the culm. And so what they believe is that each of these parts is eaten at different times during different seasons, and what they want to try to test is what is the metabolic response of the pandas to eating these different parts of the bamboo. So how are they going to go about this? Well, first, I just kind of want to review what we've mentioned in class. So this is a slide from our metabolomics chapter, and it kind of goes over like an example of how one might go about it. So you get a biological sample, run it through machine tests like NMR, which they actually used as well. And then this NMR can record the compounds you have by using their nuclear magnetic resonance. And then you can use a machine to actually help identify and compare these metabolites or other compounds that you found. And so you can better analyze them later. So some specific machine analysis that they used was, of course, like I mentioned, NMR, but they also used gas spectrometry and liquid chromatography in conjunction with mass spectrometry to get recordings of these metabolites. They also had machine analysis, which they used to compare and see if these changes in the metabolites were significant. And so the further methods continued in the study is some further specifics is there were 17 adult giant pandas. They were split into different groups, eating only specific parts of the plant. So some of them only ate shoots, some of them only ate culm, and some only ate leaves. Their daily intake was recorded, and they were weighed at the beginning of each treatment after it started, and then every 15 days after that. There were 28 blood samples taken, and these were used to analyze, of course, the metabolites and also to monitor the health of pandas to see if these were affecting how they were growing. And then, of course, there was a digestibility measure that they got by measuring the amount of food they provided, how much was actually eaten, and then, of course, how much was actually secreted. And they did it in dry mass so that there was no water changing the amount that they recorded. So here's just kind of an example of what their results will look like. So here's some NMR with a color map attached to it showing the significance of the changes between the groups. So when comparing them, so the higher ones in red show you how much more of that metabolite is in that group compared to the other one. And then in blue is, of course, lower kind of relatability. It's not as significant a difference. These are similar to kind of the machine comparisons that we mentioned at the beginning with the machine analysis. These graphs show the relatability between the two groups. So if the dots are very close together, that means that there's no real significant difference. They're all very close and found at the same spots. But here we can see they're all pretty evenly split and spread apart on these graphs. So that means that there was indeed a difference between these parts and the metabolites that they held. And so some other data that they got was their major content was this daily nutrient intake that they had. So I just wanted to point out a few odd results real quick. So of course you can see the culm group had the highest amount of intake in dry matter. If you see EE, this is just ether extract. As we know from past biochemistry, classes, there is ethers and lipids. So there's ether lipids, and these basically just refer to any free lipids that they get in the food. So if you see this, you can think EE is just fat, crude fat. It's about the same thing. But what we can see is the leaf group had the highest amount, yet it had the lowest digestibility. And we see another pattern like this in the culm group, where they had the highest intake of energy, yet they're the only group that had a net negative energy gain. So they're actually working at a deficit. And so we will kind of talk about this later in the presentation. But first, there are a few other data points that they found. Here's some metabolites that they actually found that we might recognize. So we have some proteins that we definitely recognize, valine and histidine and all those. We have some other metabolites that we've talked about, such as lactate and pyruvate. And then some that I want to put a particular focus on are these sugars over here. We have fructose, mannose, and glucose. And so what these tables mean is when you see this positive sign, that means that the former group has a higher amount than the latter group, and the negative sign is the opposite. That means the latter group has more than the former group. So what I want to kind of bring this to is our first quiz question. In this table, we see that the culm has a higher amount of glucose, but the shoots have a higher amount of these other sugars. So how are these sugars important? How do they feed into glycolysis? And what enzymes are needed for these sugars, and where can they be found? So what is the importance of these sugars? So there's that quiz question. But continuing on, what are what's the summary of these major findings? So we have the culm group. They're the highest in crude fiber, fat digestibility, of course, energy intake, calorie intake, as mentioned before, but they're the lowest in protein absorption, body mass gain, and fat intake, and then ironically enough, energy supply, despite being the ones highest in energy intake. Leaves are higher than at least the shoot group in crude fiber and calorie intake, and they are the highest intake of fat, as mentioned before, but also mentioned before, they're the lowest in fat absorption, and they're lower in protein absorption and body mass gain. Shoots are the groups that have the highest crude protein, and they had the highest body mass gain. So when pandas were measured after their diets and after eating, they actually had the most amount of growth and weight gain from eating just shoots. But they are also the lowest in crude fiber and calorie. And the paper also mentions that eating only shoots can actually lead to a long-term health risk for these pandas. And so I would like to kind of talk about some of these discrepancies and how they can relate to the things we've talked about in class. So the first one is, how does culm, which had a lower amount of fat intake, have a higher fat digestibility than the leaves who had the highest amount of fat intake? So according to the paper, the pandas have a weak lipase activity. As we know, with enzyme activity, this lipase means, lipid ACE, it breaks down lipids. If it has a weak activity, it's not going to be breaking down these lipids to be used for the panda, or by the panda. So what the scientists believe is that these leaves have way too much fat to digest. And so relating this to the McKillis-Menten equation, and also the graph here that I have to show just kind of to visualize it, this would be kind of saying that they're working at Vmax. So there is a much lower velocity, there's not as much activity. A lot of the enzyme's active site is being taken up by substrate. And so the culm groups, which have a lower amount of fat, can actually digest more of it because they're working at a more reasonable level and can actually work through it and get through the fat and digest it. And so another kind of negative with both of these groups actually is that they were low in energy, as mentioned before. So why would they be so low in energy? I mean, culm had the highest energy intake, yet it had the lowest gain. It was working at an energy deficit. Well, what the scientists believe is that since these two were both much higher in fiber is a very hard thing to break down, both physically and once it gets into our bodies. And so it's demanding a lot more energy. As we know, the TCA cycle is better for long-term energy expenditure. So if these pandas are doing nothing but eating all day, of course, they're going to be needing to use an aerobic pathway to get more energy. So they would use a TCA cycle to generate these electrons to use in the electron transport chain and give them the energy they need. So it's being put under a lot of stress to break down the fiber, which is supposed to be giving them energy, but it can't. Another thing that might be depressing the TCA cycle for these groups is that they're actually missing, or not missing, there's a lower amount of metabolites compared to these shoot groups. And so that brings me to the next quiz question. These metabolites that were mentioned in the paper were citrate. So they had a lower amount of citrate than the shoot groups. So how might this explain why the energy gained was so much higher in the shoot group, which had a higher amount of citrate, while the other ones had a lower amount of citrate? How would this affect the TCA cycle? So continuing on from that, there are still some further problems to think about with the calm, further tying into kind of this, they're at a energy deficit, how are they going to make up for it? Well, we know that there are some hormonal regulations, and so that's in the form of course, glucagon and epinephrine. So here's a picture from our slides and our book. In the liver cells, there's going to be in the body, there's going to be a lower blood glucose level. And so that will trigger this glucagon response, which will then go on to activate a bunch of regulatory enzymes. So there's pKa, which will go and phosphorylate the phosphorylase and turn it on, which will then go on to further cascade, and it'll reach the glycogen. And we're going to free up the glycogen to actually provide the glucose needed to raise those blood glucose levels. The blood glucose levels are low because we're low on energy, we're using up the glucose because we're eating so much fiber as pandas, because the culm is just so good to eat. And then similar to glucagon, we have epinephrine, which will come in and do a similar thing, but this one will try to give us a more immediate energy availability for our muscles, and trying to increase ATP levels. Instead of it just going to glucose, that glucose 1-phosphate, that glycogen will get, coming from the glycogen, will get turned to glucose 6-phosphate and then go through the glycolytic cycle and then give us that more immediate energy. And this is necessary because, again, our TCA cycle is being depressed and we need a faster way of getting the energy since it can't provide us with the amount that we need. And so the shoot group seems like it is a lot better, but there are some drawbacks. Of course, it's lower in fiber and it's lower in calories, but it also had those health risks that we mentioned earlier. So what are these health risks? I just wanted to go over them because I thought it was interesting. I know we're not in an immunology class, but we do recognize a lot of these metabolites. So from any biology class you've had, you know that if there is an increase in white blood cells and neutrophils, something's going on with your body. It's fighting something. In this case, it seems that the shoot groups are experiencing some inflammation from the shoots that they are eating. And so it is kind of harming them. It's damaging their tissues. It's not great. CRE stands for creatine levels. So the shoot groups actually have a good amount of creatine, which shows that they have a good energy reserve, which can kind of go back to what we've mentioned before, and that the shoot groups can have a lot more energy than the culm and leaf groups, which are having to use up that energy. Creatine is higher in energy than ATP, as we know, and so it's good for that kind of energy storage. But the shoot groups are very high in blood urea nitrogen, which shows a dysfunction in the kidneys, which can lead to kidney disease, and of course that can complicate health and maybe even lead to death. So we don't want these high levels. And so this all goes to show just why this study is important. So to summarize, it's very important to balance these different parts of the bamboo to actually benefit the panda and give them the nutrients and metabolites they need, and also keep them healthy. As of course we've mentioned, the shoots are high in protein. They're needed for the body mass, but you also need the energy from the culm and the fiber from the culm and the leaves, as well as the leaves provide a good amount of fat. And this also shows that in a funny way that despite the pandas evolving so far back to eat this kind of stuff, their gut bacteria has not caught up with them yet, which is a little funny that they continue to eat bamboo when they can't actually digest it. And the biggest thing is that this shows the importance of metabolomics in preservation of these species, because not only does it help us identify better ways of controlling their diet for when they're in captivity, it also helps us to get a better understanding of how their diet works in the wild. So thank you for listening, and I hope those quiz questions are not too hard.